When you think of viruses, you probably think of Ebola, HIV, or influenza, well-known villains that make us sick. But most viruses infect and kill microbes instead. These are called bacteriophages, literally eaters of bacteria, or phages for short. They all have angular heads on top of spiny legs, rather like the lunar lander that delivered Neil Armstrong to the moon. When they touch down on a bacterium, they inject their DNA and turn the microbe into a factory for making more phages. These eventually burst out of their host in fatal fashion. Phages don't infect animals, and they far outnumber the viruses that do. The trillions of microbes in your gut can support quadrillions of phages. A few years ago, Rauer's team member Jeremy Barr noticed that phages love mucus. In a typical environment, there will be 10 phages for every bacterial cell. In mucus, there will be 40. The same fourfold spike in phage concentrations exists in human gums, mouse guts, fish skins, marine worms, sea anemones, and corals. Imagine hordes upon hordes of them, stuck head first, their legs outstretched and waiting to embrace passing microbes in a lethal hug. And these mucus-bound phages might be more than just crude tools for killing microbes. Rauer suspects that animals, by changing the chemical composition of their mucus, could potentially recruit specific phages, which kill some bacteria while providing safe passage to others. Perhaps this is one way in which we select for our favored microbial partners. On page 129 of the Superhuman Organism, Dieter says, virtually every NCD has unhealthy inflammation with excessive oxidation at its core. Oxidation is a normal chemical reaction that utilizes oxygen, but some of the byproducts of oxidation are free radicals that can damage our cells and tissues and alter our proteins and DNA. Free radicals of both oxygen and nitrogen are useful if you are trying to kill pathogens. They are harmful if our own cells encounter them. That is one of the reasons to eat a diet filled with antioxidants and sometimes take antioxidant supplements. We need to avoid oxidative damage of our own cells, but inflammation usually produces oxidation, and unhealthy inflammation produces so many free radicals that it is impossible to scavenge them all with antioxidants. When this happens and our tissues receive years of oxidative damage, they either lose tissue function or the tissue cells become cancerous. Inflammation, as nature intended it, is both necessary and useful. However, it needs to be 1. Appropriate for the task, 2. Directed at the narrowest target possible, the pathogen, not an entire organ, and 3. Resolved once the legitimate threat has passed. This section is taken out of Missing Microbes by Blazer on page 138. The green is paraphrasing and the yellow is direct quotes. It says, the inside layer of the digestive pathway is covered with dendritic cells and lymphocytes, which act like sensors detecting and reacting to bacteria. Cultures of lymphocytes give rise to B cells and T cells. Some T cells increase inflammation, while other kinds regulate or inhibit a particular immune response. There are fewer than 100 species of bacteria that cause infectious disease in humans. By contrast, the thousands of species in our guts are mostly harmless. At worst, they are passengers or hitchhikers. At best, they are invaluable parts of our bodies, not takers of life, but its guardians. They behave like a hidden organ, as important as a stomach or an eye, but made of trillions of swarming individual cells rather than a single unified mass. The microbiome is infinitely more versatile than any of our familiar body parts. Your cells carry between 20,000 and 25,000 genes, but it is estimated that the microbes inside you wield around 500 times more. This genetic wealth, combined with their rapid evolution, makes them virtuosos of biochemistry, able to adapt to any possible challenge. They help to digest our food, releasing otherwise inaccessible nutrients. They produce vitamins and minerals that are missing from our diet. They break down toxins and hazardous chemicals. They protect us from disease by crowding out more dangerous microbes or killing them directly with antimicrobial chemicals. They produce substances that affect the way we smell. They are such an inevitable presence that we have outsourced surprising aspects of our lives to them. They guide the construction of our bodies, releasing molecules and signals that steer the growth of our organs. They educate our immune system, teaching it to tell friend from foe. They affect the development of the nervous system and perhaps even influence our behavior. They contribute to our lives in profound and wide-ranging ways. No corner of our biology is untouched. If we ignore them, we are looking at our lives through a keyhole. All of the highlighted paragraphs that I have on this paper is from a book called Missing Microbes by Blazer. I don't have the book because I read it when I was in college, but the first one I'm going to read, it says, during the Human Genome Project, beginning in 1950 and ending in 2003, it was discovered that the human genome contains between 22 and 25,000 genes. This is a very small amount of genes, considering that 99% of the unique genes in your body are bacterial and only 1% are human, on page 35. And the Human Microbiome Project, which began in 2008, different from the Human Genome Project, uncovered tens of millions of microbial genes. And just to clarify, the green parts is me paraphrasing what was in this book, and the yellow part is a direct quote. So on page 82 of the Superhuman Organism, it says, in 1950, Cornell-trained geneticist Barbara McClintock showed that genes could be mobile and could indeed jump and change locations, at least along chromosomes, within a cell's nucleus. It took decades before her revolutionary Nobel Prize-winning discovery was fully embraced and appreciated. But if genes could do that, could they jump between species? An early result was reported in the 1950s showing this was possible among bacteria, including the one that causes the disease diphtheria, Corynebacterium diphtheria. In the case of the diphtheria-causing bacterium, genes that were transferred into the diphtheria bacterium by bacterial viruses called bacteriophages controlled how aggressive or virulent the bacterium was in producing the disease. Following this, it was shown that the genes providing resistance to antibiotics could be horizontally transferred or swapped between different species of bacteria with the help of the same viruses. It turns out that our bodies are perfect locations for horizontal gene transfer. In fact, the microbes within our microbiomes are known to use locations such as the gut as a type of swap meet. We only recently discovered that different bacterial species living inside us in the same body location can occasionally exchange genes. But can this type of transfer happen when the recipient is a higher organism, a plant, an animal, or even a human? And what they found is that that is the case. And then on page 83 of the superhuman organism, it says that tens to hundreds of foreign genes of probable microbial origin have been identified in the human mammalian genome, and many of these seem to code for proteins with unique enzyme activities. And then towards the bottom of the page it goes on to say a prior swap of genes from microbes to our ancestors' mammalian chromosomes would mean that some of the approximately 1% of human mammalian genes are not really mammalian at all. At least some of those genes sitting on our chromosomes today were swapped into us from microbes. So the more we look, the less of us is actually non-microbial and free from microbial influence. If these once microbial genes help us to do useful stuff that we could not do before we grabbed them, that stuff we now can do is of microbial origin, even if the capacity resides in our own chromosomes. It was found that in one study, Europeans had different numbers of bacterial genes. A percentage of over 70 of Europeans in the study had roughly 800,000 genes. The smallest group, a percentage of a little over 20%, contained roughly 40,000 genes. The group with the lowest amount of bacterial genes were found to be more susceptible to obesity. On page 35 of Missing Microbes, it says, another study showed that mice treated with antibiotics were roughly 15% fatter than mice left untreated. Low doses of antibiotics altered the normal mixture and abundance of microbes in the intestines, therefore changing the normal functionality of microbes. In some circumstances, bacteria in the colon can ingest leftover remains, creating short-chain fatty acids that are absorbed in the colon. SCFAs can make up to 15% of calories in humans. In this experiment, it was found that the liver upregulated genes needed to make and transport more fat. On page 85 of the Superhuman Organism, it says, being able to control when a gene is turned on, how much of a gene's product can be made, and when during development the gene is on or off, can be the difference between life and death, health and disease. Consider the production of hemoglobin, the oxygen-carrying protein in blood. Without adequate oxygen, your cells and tissues would die. It turns out you have different types of hemoglobin, and they are tailored precisely for different life stages and the oxygen needs of your tissues during those specific life stages. The production of embryonic versus fetal versus adult hemoglobin is under the control of epigenetic gene switches. The switches are flipped at precisely the right developmental stages for everything to work. It turns out that one of the small molecular metabolites of gut bacteria, sodium butyrate, can control these gene switches and affect hemoglobin expression. On page 118 of the Superhuman Organism, Dieter says, when it comes to microbes, just as with environmental chemicals, here's a prescription for good health. The right place, specific body location, the right amount, dose, the right time, developmental menstrual cycle or circadian cycle timing, and compatible with our mammalian self. The flip side is equally a path toward pathology and disease. The wrong place, the wrong amount, the wrong time, or incompatible with our mammalian self often causes significant health problems. On page 117 of the Superhuman Organism, Dieter explains how a wonderful and health-promoting discovery uncovered between the years 1979 and 1984 may, in the long run, end up being viewed as a tipping point for the fallout over the 20th century war on microbes and overuse of antibiotics. Barry Marshall, the Australian physician, and his collaborator Robin Warren reported the link between a spiral-shaped bacterium only found in humans named Helicobacter pylori, H. pylori, infections of the stomach, and peptic ulcers. Their report was first published as letters in 1983, with a full report in 1984 published in the medical journal Lancet. So these scientists found a link between a bacterium called Helicobacter pylori and stomach ulcers. And so why might you want some H. pylori, which in some circumstances can produce peptic ulcers or stomach cancer? Because purging the body of H. pylori is equally associated with other NCDs, and we have a good idea about how the mechanism of how this happens. It turns out that the persistent presence of H. pylori helps the immune system become more tolerant and reduces the risk of asthma, allergies, and inflammatory diseases such as inflammatory bowel disease. Beginning with immune cells called dendritic cells that sample the environment, partly through H. pylori's effect on dendritic cells, there is an increase in the maturation and numbers of regulatory T cells called natural tregs, and these are critical to avoiding the inflammation that supports numerous NCDs such as asthma, allergies, and irritable bowel syndrome. So then there was these two scientists, Dennis Casper and Richard Blumberg, and they was wanting to find, these groups used the C57 Black 6 strain of mice, which is a standard research model for immunology, to investigate the effects of commensal bacteria and their metabolites on early immune maturation and susceptibility to later life non-communicable diseases. And the disease was colitis, which is similar to ulcerative colitis in humans, one of the two parts of inflammatory bowel disease. In this strain of mice, the lack of commensal bacteria makes the mice highly susceptible to colitis when they are exposed later in life to oxazolone. So they found that the immunological mechanism for this colitis is well described, with specific immune cell populations and immune hormones leading the way to produce disease. The dysfunctional immune process in mice appears to be similar, if not identical, to that which produces human ulcerative colitis. So basically they asked a question. They wanted to figure out if there was a microbe that could combat ulcerative colitis. So their first question was whether a single commensal gut bacterium could protect against later life colitis. The answer was yes. And the bacterium that provided resistance to the disease was Bacteroides fragilis, or Bacteroides fragilis. This bacterium is rod-shaped, does not need oxygen to grow, and is usually one of the immune system's friends as long as it stays put in its region of the gut. The Harvard researchers then went a step further and asked if there was a critical developmental window when the bacterium had to be in the newborn mouse to produce resistance to colitis. The answer was it had to be there by 1 week of age. After that, the addition of B. fragilis could not prevent colitis. Next, they asked how the bacterium made the mice resistant to disease in the newborn. The answer was it blunted the proliferation of a specific population of gut innate immune cells called invariant natural killer T cells, iNKT cells. In the absence of the bacterium, these cells had a burst of proliferation in the infant mouse, and that burst was what made the mice susceptible to colitis for the rest of their lives. If the bacterium was present in the gut, the burst was significantly lessened, and the mice were resistant to colitis for the rest of their lives. It was a remarkable finding about the importance of an early developmental window for the immune system and the need for the microbiome to be in place to avoid later life disease. Finally, the researchers asked whether the entire bacterium was needed or if a metabolite of the bacterium could produce the same beneficial effect on the developing immune system. They found that a particular type of lipid made by B. fragilis, given at just the right developmental window, could suppress the amount of iNKT cell proliferation and make the mice resistant to colitis. This is one gut bacterium, one immune change, and one NCD. Imagine the opportunities to reduce the prevalence of NCDs when the entire microbiome can be effectively managed to support the best possible cultivation of our immune system. So now I want to kind of get some insight into this, because on one hand you got Helicobacter pylori, which trains the immune system and shows it how to upregulate inflammation or downplay inflammation, but if there's an imbalance and there's too much of it, can cause ulcerative colitis. And then on the other hand, and Helicobacter is a helix shape. And then you got Bacteroides fragilis, which is a rod that regulates the negative side of Helicobacter and prevents ulcerative colitis. So one is a rod and one is a helix, and they're both bacteria in the gut. And if you put them together, I'm not saying that they go together, like they're probably in two different places in the gut, but they balance each other out, creating health. So, but if it was put together, it would be a helix around a rod, just like nucleotides go around a phosphate backbone in DNA. Just like the goddess Hermes had the rod with wings with the two serpents around it that he got from Apollo, that was known for medicine and healing. Or the goddess of Asclepius, who had the single snake around a rod or staff, that was the goddess of healing and medicine. And even in the Bible, it talks about the rod, and Moses threw down his staff and it turned into a serpent. So you have these two bacteria in the gut that put together is the medical symbol for health.